Good morning, everyone. Today we will be going a step ahead again. So I believe that in my previous two classes you have got a clear idea that how structural elucidations can be done in a very constructive and in a very methodical way, how we can understand small details of the entire process, and then based on that what are the best tools which we can use to elucidate the structural integrities of your sample of interest. So taking a step forward in that direction, today we will be again understanding, or rather focusing on how to understand a crystal, partly of the lattice structures and everything which we have seen in a previous session. But today we will be focusing on crystallization process, that what are the methods, what are the parameters which actually affect the crystallization process, how you can achieve a good crystal, because until unless you get a proper crystal, your entire objective of this study cannot be fulfilled. So you need to get a proper crystal. And why getting a crystal is important, because once it is a crystal, the structural orientation of the internal atoms becomes very finite and becomes very definite in shapes, and then we can narrow it down to unit cells, where we can understand that how this unit cells diffract the X-rays coming onto them, and based on the pattern of diffractions we further study the distance between the interatomic distance between them, the angle of diffractions, whether these diffractions are constructive or destructive in nature, and obviously the electronic cloud which these X-rays are experiencing. So we have gone through the protein structures and its basic you can say the integration or the basic architecture of it, and also we have gone through how these crystals and the orientations, or basically the packing of the crystals, look like. So from a single or a simple type of crystal to a more and more complex type of crystals. But the best or the advantageous part is these crystals will have a fixed orientation which are repetitive in nature. For example, if you see, this is the one block of this crystal, and if you keep on adding this block, it is repetitive in nature, and that is where we have an advantage of identifying a unit cell and then studying them. Now today without taking more time, rather we will be seeing what are the major steps in crystallization. We have understood about the patterns or the packing architecture of the different types of crystals, and once you have understood this, what we have to do, we have to get crystals. So how you can achieve or how you can get crystals in your sample. So these are the major steps of crystallization. The main objective is to achieve a crystal of the protein which should be as pure as possible, and if your protein is crystallized, a proper crystallization has happened, then the structure, the lattice orientation, the interatomic distance, everything will be in a fixed conformations, which will give us an advantage to study with X-ray diffractions. Now to achieve this, what you have to do is we have to select a lot of buffers, protein buffers, in which the protein has to be soluble first, and then slowly by various processes how this buffers will slowly evaporate and move away, leaving behind the crystal in its actual true form. Now this is also possible by bringing the protein solution to supersaturation a stage, or a supersaturation stage, where you see there have been lot of spontaneous nucleations and seeding of new crystals being taking place. Okay, and I request all of you, please mute your microphone unless you have any questions. Okay, so, and the last is, yes, you have not only to seed or start the crystallization process, but that crystal has to keep growing so that you get the full crystal from the entire droplet which you are, or the protein solution which you are using it, right? Now this may sound very easy, because I may say, okay, just you add the solution of the protein and then you evaporate it. After evaporation, the protein crystallization will take place and you can use it for your X-ray diffractometer. But that is not as simple as it sounds right now. Why? So we have to understand step by step. The first parameter is solubility. Okay, now solubility we know as and when we have taken a solvent on which some solute or some salt is being added. So initially when you keep adding, what happens is, it will not get saturated, because the solute, or basically what you are adding onto your solvent, that concentration is very less. So you can add more and more, and gradually if you keep on increasing the salt onto this solution or onto this solvent, basically what happens is, so the amount or the capacity of dissolving of this salt gradually decreases, right? So what we see, initially the protein solubility will increase, and after a certain stage, then what will happen, it will begin to decrease when the salt concentration is very high enough. And you see in this plot, you see the ionic strength, and in this ionic strength you see the plots here of different salts. And for most of the plot, you see initially you see a steep rise, and then slowly what happens, it's getting saturated, and at times the solubility also falls. So for others, some salts, you see it is not saturated, it is even falling back. Okay, so solubility decreases. So we have to understand what should be the concentration of protein you are mixing in the solution, right? And if it is getting saturated, if it is getting undersaturated, oversaturated, those things you have to understand how much solubility capacity of your particular protein of interest where you want to study and you want to crystallize, that you have to see, and then based on this how much you want to add. So this concentration regulation is very important. Second most important parameter is supersaturation. Now supersaturation is, saturation we all know, it's getting saturated, there are no more space. But what is supersaturation? So if you see this diagram where you see the protein and the salt, and if you try to plot it, the concentration of protein and based on the concentration of the salt, and see the saturation in a very schematic or in a very representative manner, then it is like supersaturation is a stage or is a phenomena which is achieved by adding more of a substance, okay, than normally be dissolved. Means if in my 1 ml solvent, usually suppose 1 milligram of the substance can be added, if I add more than that, and if that condition will be a supersaturation stage. Okay, now why this is important? Because at that stage what happens is, the entire mixture or the solvent along with the salt mixture, that becomes thermodynamically unstable. And once it becomes thermodynamically unstable, what will happen, the crystallization process will keep starting or initiate, right? And that is the most important, you can say, the phenomena. But it can again be, so when you have a droplet, okay, and it is that solution or the protein solution, and by the process of slowly evaporating the solvent from that solution, so there are 3 layers or zones which usually is observed. The first one is zone 1, which is in green, you can see here. In the zone 1 is called metastable zone, right? So in the metastable zone, what happens, the solution may not nucleate, okay, for a long time, but this zone will sustain the growth. So it is frequently, or it is very essential to start or to maintain the seed crystals. So this seeding of the crystals will start from this zone 1, okay, But again, you have to understand this is a thermodynamically stable zone, so you do not see a very fast changes or nucleation process. Okay. The second zone, that is the blue zone. The blue zone is the nucleation zone. Now what is nucleation is? Where protein crystals nucleate. Okay, so I'll come to the nucleation process also in the next slide. Where the protein crystals, each of these units you see started forming and then keeps growing. So this is the zone of the blue zone, where you find the proteins have started, or the crystals have started nucleating and gradually growing, right? The third and the red zone is the precipitation zone, and in this precipitation zone what you see is proteins do not nucleate but precipitate out of solution. Now precipitation is when the final crystal deposition have already started, and nucleation is a step ahead of it. So nucleation and then followed by precipitation. So in the last zone, that is the red zone, actually the crystals which we are looking for will be observed. Okay, so again there are three zones, and in these three zones, first one is a metastable zone, where you see it is in the green color represented, and it is more stable and it will nucleate for a very long time, and you see the evaporations is happening from this surface only, but this will initiate the formation of seed crystals inside. The second one, zone 2, is the nucleation zone, where the nucleations will start and the protein crystal nucleate will start forming, and gradually it will keep growing. And the third one, the red one, again is the precipitation zone, where the crystal precipitation will take place. Okay, so this is the entire, you can say, zone wise distribution. Now what is nucleation? So I was talking of nucleation. Now this is a phenomena where, or you can say it is like dust like particle, or basically nucleus, a tiny, very tiny seed crystal, okay, a small protein aggregate which starts the process of crystallization. So to start the entire process of crystallization, what happens is, first of all, it is a solution, or it is basically a mixture. The protein is in a solvent, and this is the first step where the unit or the crystallization process have started, right? And this is as small, so you can see this crystallization or the nucleation is or a protein crystal nucleation will be comparable to a very small fine dust particle. It is as small as that. It is not a crystal, but the crystallization process has started in the nucleation process. Now the common problems which you find is, if the supersaturation is too high, that is why the solvent or the saturation part is very important. What is the pattern of how much protein can be dissolved in the solvent, that is, understanding that is very important. So if saturation is too high, too many nuclei will form, and hence an overburden of tiny crystals. Now what will happen when in a small area you have too many tiny crystals? The uniformity and the uniformal growth of all these unit crystals will not be there, and what will happen is they will squeeze into each other's space and giving an non-homogeneous crystal structure. So the crystal structure will not be homogeneous, and then it will not be suitable for your X-ray diffraction studies. So this is very important, that your concentration in the supersaturation state should be optimum, so that the nuclei or the small nucleated crystals, tiny crystals, they have enough space and they can grow uniformly. Okay, if supersaturated solutions does not experience spontaneous nucleations, it does not experience crystal growth, and only occurs in the presence of added nuclei or seeds, then also it is a problem. Because if it is not a spontaneous nucleation, what will happen? The uniformal growth or the uniform directional, or in all the 360 degree, the development of the crystal will not take place. It may be in one side, it may be like the growth is happening in one particular plane, and in that plane you see the formation of the crystal is happening, but in the other planes they are not doing it. Until and unless it is a 360 degree spatial uniform crystal development, again again the problem remains, and we will not be able to use that type of crystal for further structural studies. So nucleation, supersaturation, and solvent, how much solvent or solubility of that protein, these things are very, very important when you are preparing or when you are starting with your crystallization. Another very important is, so keeping in line with the previous, like the growth I was talking of. So this is a small example. You see here in this example, this has been done in University of Toledo, where they have shown that unit growth or unit seed growth of the crystal may not be always the true scenario or the true case with all the crystal growth development process. So what happens is, in this case you see a unit of 4, and when you turn it, you see the, there is a height, there is a width, there is a length associated with this, and this particular crystal growth is happening with basically 4 small unit cells coming together, and this quadrant is slowly doing. So this is basically the growth units of lysozyme, which I'm showing it here. And in this growth units of lysozyme, you see the step growths or the growth units, they are 4 unit. So basically it is not a single small unit which you see here, but it is 4 together, and this together work and then keep on growing and making this a nucleating lattice. So this lattice formation is taking place. So it is not always that a unit cell will grow on its own. You can have a multiple of this, but the idea again remains the same. The growth pattern should not change. You see here the growth pattern is uniform in all the directions, right? And this growth pattern should be uniform in all the directions, so that when the X-rays fall, the diffraction patterns can be used to study them. If the internal pattern is not uniform, then it makes no point. We cannot decipher information. We cannot get information from the diffracted X-ray light. Okay. So the growth of this crystal can stop in between, and they can be developed because of several defects also. So the one major thing is, if the thermodynamic equilibrium, that is what we have been talking of, if the thermodynamic equilibrium is not maintained, the process will stop. The common factor is the solution in which the crystal exists. This is often not the same as the original crystallization screening solution, but is instead the solution that exists after some degree of vapor diffusion. The equilibrium through the dialysis or evaporation is not maintained. So as I said, if the equilibrium is not maintained, the growth will stop after a certain initiation, and then the crystal development will not take place. So you have to also see, yes, your nucleation has started, the crystallization process has started, but it should keep growing uniformly and it should not stop in between. Now, this is very important. What are the major factors that affect your crystallization process? First and foremost is the purity of the proteins. So whatever protein you have extracted, it should be 100% pure. Why? Because if your protein is not pure enough, what will happen? The intermolecular gaps will be filled with a non-protein molecule, and then the distance between them will vary, and that will lead to a irregular crystal formation. So again, irregular crystal formation is what we are not at all looking for, and that is why you need to have a very, very pure protein. And as and when isolated, there should not be any sort of contaminations either with any sort of metal ions or anything else, and then this protein should be dissolved in the particular solvent or the buffer and then used for protein crystallization. Second, yes, we already discussed about the concentration, because if the concentration is less, then it will not reach to the supersaturation point very easily. If the concentration is more, then it will reach to the supersaturation, and there may be chances of too much seed crystals in a very small area, so that may lead to non-uniform crystal growth. Okay, so the concentration of the protein is very important. Then the starting conditions, or basically the makeup of the protein solution, how you prepare the protein solutions, in what ambient temperature, pH you are keeping it, although pH and other things you'll be taking care of is coming. So what the point is, it has to be as uniform as possible, and as you have to maintain it also throughout the process. Then comes obviously the agent or the reagents. The reagents will be like whatever the precipitating agent you are using it, what is the concentration of it, what is the grade of it in terms of purity. So if there are any impurities in that also, then also it may affect the process of precipitation. Obviously temperature is very crucial, because the temperature will assure the slow and steady evaporation of the solvent, and leading to the formation of nucleation and then further to crystallization process. Okay, so if your temperature, suppose I have started at 4 degrees today, and tomorrow all of a sudden it is changed to 24 degrees, and then again the temperature fluctuations has happened. So this will lead to non-uniform evaporations, and the crystal structure will be affected. So temperature is very important criteria here. pH, yes, the pH has to also be maintained, because based if there is any isotonic solutions, because the solvent and the solution will have that difference, so pH will maintain what will be the rate of evaporation again, because based on the isotonic solution and surface tension. So those things will also be playing. And last but not least, the additives which you are using, the cofactors, the detergents which you are using, those things also has to be taken care of. So again, coming back to the purity of proteins. The purity of proteins, yes, it has to be as pure as possible, so that your entire thing is as homogeneous. Basically I'll say the structure should be as homogeneous as possible. Now partial proteolysis products are also there, so partial proteolysis may happen, and because of that partial proteolysis of the protein, the protein formation will not be accurate, so this has to be taken care of. There may be cases of oxidation of particular amino acids, so that can also happen. So oxidations process. Another is amidations, is a process where you see changes in the structure. So this asparagine so can change and have a deamidation. So amide is a functional group which keeps changing, or which may change in the process, and that may lead to change in the amino acid sequence. So this process will also has to be taken care, otherwise the sequence of the amino acid of the protein will change, and that will lead to non-homogeneous structure. There may be some post-translational modifications, so during the process of translation and after that there may be some modifications, so that has to be also taken care, so that it doesn't affect the purity of there. Oligomerizations may happen, so if your protein have a tendency of having multiple chains, then oligomerization process may start, and your purity of the protein may not be there, so you do not actually get that structure. Your protein may also have a tendency to go for isoforms. It can be a misfolded populations, and the structural flexibility. So all these basically are structural flexibilities, and because of structural flexibility, what will happen is the conformation will not be fixed, and that is why whenever you are getting a crystal, again the lattice or the crystal structure will not be homogeneous in nature, and you will have problems in understanding the structure. Then concentration, yes. So concentration, why? Because if you want to have a repeatability in your experiment, the concentration has to be very there, it has to be consistent, and these are the major issues, because you should have a reliable assay to check this out. So there are two assays, like you say extinction coefficient for tryptophan, and you can also use a Bradford assay as a standard, and then this too can be used to see whether, because see, once you do crystallization, this is not a one-time process. If anybody wants to repeat it, so your process should be as good as it can be reproduced by any other person who follows the protocol as religiously as possible. Okay, So E. coli expression systems are for crystallographers are commonly used method for obtaining a protein, and the problems can also arise because of low expression yield, and this can happen. So why the expression of this protein yield is low? Because you have a cytotoxic environment which is actually killing the E. coli. You may have unstable plasmids or mRNA. You may have that misfolding because your structural stability is not there in the protein, so you may have misfoldings, and some common eukaryotic codons are also available or at found, and they are rare in E. coli, but still at times you see some of this happening, and which may alter your protein concentration which you're trying to do it. So these are the practical problems which I am discussing with you. You should always keep this in mind while going through this process. Okay, so now the main point is to know that your starting conditions for purposes of reproducibility are set up. Okay, so your starting conditions are set up. Now you should focus on the reagent part. Yes, your precipitating reagent, as I said, there are several types of salts, organic reagents are being used. So what are the reagents you are using? What is the concentration of the precipitant you are using it? Those things has to be taken care. Again, coming to the temperature, yes, temperature, because the dynamics of how protein solution reaching to its super saturated state has to be understood, and that is entirely controlled by the temperature. So the easiest are 4 degrees centigrade and the room temperature, and also you can try something around 12 to 15 degrees. So these are the temperature ranges. You can either try at 4 degrees or at room temperature, and also in between like something around 12 to 15 degrees if you keep it, and this temperature will maintain. And you just, so rather I think I've seen in most of the labs they prefer to keep it at room temperature, but again the process of crystallization is something which you cannot predict, because your crystallization process may happen in hours, in days, in weeks, in months, and even in years. Yes, you are hearing it right. It may take even 1 year, 2 years time also to have a crystallization complete. So you cannot predict the timing, and based on that, the little or the minimal fluctuations which your entire setup is experiencing, the better it is. Okay. And lastly, not lastly, again the pH. Yes, the crystal packing, as I was saying, because this crystal packing is referring to the arrangement of the atoms or the molecules in the space. So this arrangement of these molecules in the space is a spatial arrangement, and how they are related to each other in that particular space in a fixed conformation. That crystal packing is very, very important, and that is influenced by the surface charge, and which is again depends on the pH. Okay, so if you have hydrophobic interactions are less important than electrostatic interactions in crystal packing. Okay, this is why pH is very important. And last but not the least, yes, the stability of the protein, the homogeneity can be disturbed with the additives. Okay, but suppose after doing all this exercise still you are nowhere near to getting a perfect crystal, or not even the crystallization or the nucleation process has not started. So what you have to do? So you have to see new constructs. Yes, you have to try for deletion mutants. You have to see for complexes with the substrates, what is the complex it is forming. You have to see the protein complex with fab fragments. Okay, so these are basically fragments, small fragments, and what is the protein complex which you are working with, and obviously the homologous proteins availability. So how homologous proteins are there, and what is the structure, what is the orientation of. So you should try to understand these basic differences before you start, and I think this will help you in having a better and going towards the formation of crystallization. Now these are the previous setups, or the previous do's and don'ts before you start your experiment or when you plan your experiment. But the thing is, what are the methods? We should also understand how you can form crystals, right? So grossly there are three which I will be discussing today. One will be a vapor diffusion, another will be a slow evaporation, and the third is dialysis method. Okay, so the first one is basically a vapor diffusion technique, and in the vapor diffusion technique there are basically two types. The first one is a hanging drop vapor diffusion technique. So if you can see here, in this is basically a cover slip, and this is siliconized glass cover slip, and on this siliconized glass cover slip you have one drop of the protein solution. Now this protein solution, once you drop it, you put this cover slip upside down. So you see this protein solution is hanging. Okay, so this is that is why it is called a hanging drop. So this protein solution is hanging, and in this chamber basically you have the crystal screen buffer. So this is basically a well where you have the solution buffer or the crystal reservoir buffer, and this reservoir buffer has been taken in like from 0.5 to 1 ml, and you are taking a drop, just a small drop, which will be hardly 5 to 10 microliter mul, and you will add the same amount or the same volume of this crystal screen or the reservoir solution and add it with this. So suppose you have added 4 microliter of this, or suppose you have added 6 microliter of the protein solution, you will also take 6 microliter of the reservoir solution and add to it. So the resultant drop volume will be 6 plus 6, 12 microliter, and then you place it in this chamber upside down. Okay, so the cover slip is inverted over the top of the well and sealed with vacuum grease. So you see here, this is basically the grease. So this is now a closed environment, and in this closed environment what will happen, slowly the solvent from here will start evaporating, and this will reach from a solution to a super saturation stage, and at a super saturation stage the crystallization process will start, and gradually you will get the crystal. So the precipitation concentration in the drop will equilibrate with the precipitation concentration in the well via vapor diffusion. Now these concentrations will be equated or equilibrated by the process of vapor diffusion, and that is why it is called a hanging drop vapor diffusion technique. Right, so it is very simple. You have to take a siliconized glass cover slip, on that add a volume of solvent, protein solution basically, add equal volume of the reservoir solution to this drop, and then put it upside down in this closed well or reservoir and airtight it, seal it with grease. Keep it at room temperature, and gradually what will happen, the precipitation concentration in the drop will equilibrate with the precipitation concentration in the well by the process of vapor diffusion, and then the crystallization will take place. Right, next, it is a more or less very similar technique, but somewhat the setup is different. Here, instead of having a cover slip on top, what you're doing, you're using a small platform in the middle, and on this we are using a same drop of protein solution added with the equal volume of the reservoir solution, and then you finally leave it with the closed environment. Now why we need to do this? Because if you want to have a larger crystal size, then this is where it is there, and at times your protein solution, the surface tension may be less. So while in the hanging drop or the hanging drop technique, due to low surface tension, this drop may fall, and that is why if you want a larger crystal, if you want to increase the volume, you can have that. And you can see here the sample size here, 20 to 40 microliter, which was earlier 6 to 10 microliter. Now you can increase the volume to 20 to 40 microliter. Rest of the process is as same as the hanging drop technique by the method of vapor diffusion. Okay, then comes oil immersion micro batch. Okay, so this is basically a multi-well batch. You can see here multi-well plate, and in this you have lot of wells on which you are using an oil immersions, right? So you're using paraffin oil basically in this case. What is happening is this has become a very handy technique because you can have multiple of these wells, and each individually you can get or use them to get crystals. See, crystallization is very uncertain. You don't know about the time frame, you don't know about actually you have done it, so it is like, you know, it is like shooting in the dark. So if you have more and more wells or where you're trying, the chances of getting a crystallization will increase, and that is why over a period of time this method has increasingly become very, very popular. Okay, but your typical sample size will be again less. It will be 1 to 6 microliter, but it doesn't matter. In X-ray crystallography the smaller size doesn't really matter. You want to get a lattice crystal, so that is the entire idea. Now here you can see here in the first one, paraffin oil, it allows for little to no diffusion of water through the oil, and this is a batch experiment. As I was saying, you have multiple wells, so in a batch of number of wells you can do it, and all at the specific reagents are present at a specific and relatively unchanged concentration. So when you are doing a batch experiment, it is easy to do it in a series of wells, and then you see and observe it. So it may, if you do single, the chances of getting crystals are again less. In a batch experiment it might improve, so your probability increases, and that is why the popularity of this particular method has increased over a period of time. Now in the second one, you see the oil is there, and it is in 1:1 mixture. The silicon as well as paraffin oil 1:1 is there, and again in this case also it allows a evaporation, and this is again slow diffusion evaporation through the oil. So this is basically an evaporation method, and the concentration of the protein and reagents in the drop does not on the other hand increases. The concentration of the protein and the reagents in the drop does increase over a period of time. What is happening? You are not closing this particular wells, and that is by evaporation slowly is happening, and the concentration of the proteins and the reagents in the drop slowly differs over a period of time. Okay, now the third one is microdialysis. In microdialysis, again this is a similar reservoir setup which we showed, or which we discussed in the previous slides. Here interestingly we have a microdialysis button. So this is one small platform, and in this microdialysis button is there, and inside the sample is there, and it is dipped in the reservoir solution, and again airtight in a closed environment. You are keeping it sealed with a sealing tape or cover slip. Now the dialysis buttons can be, you can procure it from the market, and your sample size in this case can be of varying size. Like it can be as small as 5 microliter, it can be as big as 350 microliter, and that is where you have the flexibility of having smaller as well as bigger crystals. Now in the dialysis experiment, what you do is the sample is introduced. This is the sample which is introduced to high salt concentration within the button, and that are allowed to equilibrate with lower salt concentration in the buffer. So high salt concentration is here, low salt concentration is here, and it is allowed to equilibrate over a period of time, and this process is called salting in method. Salting in method. It exploits the fact that not only does protein solubility tend to decrease with very high ionic strength, it also has a minimum at very low ionic strength. Okay, so what the process is, you have this low concentration here, high concentration of salt here, and by process of salting in, slowly the crystallization process will start, because it will try to equilibrate, and you'll see a trend decrease in very high ionic strength and on a minimum at very low ionic strength. So this trend will keep on happening, and gradually crystallization process the crystallization will start. Okay, so we have certain tools by which we use, or we can see the crystal mounting is being done, and these are basically the capillary tubes, the glass or quartz. You can see these tubes are used for crystal mounting, and these are equally important because if you are not properly handling it, if you are not properly mounting the crystal may get damaged also. So after all that hard earned actually getting the crystals, it should be handled. So you also have a cryo loops where you maintain the temperature also is possible with a thin nylon. You can see the loop like structure they are used to mounting your crystals. Okay, so I think today I'll stop here, and next class I will start with the problems in X-ray crystallography, basically with the phase problems, and then we'll conclude with the X-ray crystallography technique. So my idea is, like, we have understood what the X-ray crystallography technique is. We are using, or we are manipulating the properties of X-rays to decipher the structural things. But again, crystal is something which is very important, so crystallization of protein has to be done, and these are the methods of protein crystallization which you can achieve in your lab. Once you achieve a proper crystal, then you can take it to your crystal or diffractometer, and then you study. And how you are going to study it with the terms of contortions, torsion angles and everything, I will take it in the next class.